In this video, we will discuss about the soluble E-Cadherin effects on invasion and aggregation of the cells. So, whether this soluble E-Cadherin, which is found to be cleaved, or which is found to be the cleaved off cellular part of this E-Cadherin, whether it affect the invasion and aggregations of this pancreatic cancer cells, that we will discuss this, and the pancreatic cancer cells that they have used to check any role of the soluble E-Cadherin is PANC1 cells. So what's the experiment that it did is they first treat the cells, pancreatic cells, that is BXPC3, with and without HK7. As we know that from the previous experiments, that when they treat it with in the culture with HK7, it will lead to the excessive cleavage of these E-Cadherins, and thereafter they go through the 2 cycles of immunodepletions to remove or to neutralize and remove this soluble E-Cadherin on one case, and on the other case they have this soluble E-Cadherin. And the results after this first cycle is shown in this lane 2 and lane 3, where we can see that when they used this anti or alpha-E-cadherin to neutralize this soluble E-Cadherin, they are still able to see some amount of this soluble E-Cadherins. However, after second cycles, they can see complete removal of this fragmented E-Cadherin or the soluble E-Cadherin. So moving forward, they used this second conditions, supernatant, for their further studies. First, they checked the impact on the invasions. So in the case where there is no soluble E-Cadherins are present, they are able to see a reduced invasions. And panel B and panel C results are for the PANC1 cells. So what it is, they took the PANC1 cells and cultured them with the supernatant from the second cycle of this immunodepletions. So in the case of the case where there is no soluble E-Cadherin presence, they are able to see reduced cell invasions compared to the case where the soluble E-Cadherins are present. This could indicate that this, because of the absence of this soluble E-Cadherin, it will leads to the suppression of invasions, as this E-Cadherin plays a role towards the cell invasions. E-Cadherin plays a role towards the cell adhesions. So in the absence of this E-Cadherin, the soluble E-Cadherin, the excessive adhesions will not occur, and this will in the absence of the soluble E-Cadherin, the problem in these adhesions will not occur because on the ECM, only the E-Cadherins that are present on the cell surface or the baseline soluble E-Cadherins are present in the PANC1 cells will compete for the sites on the sites on this ECM, and these experiments are done on the ECM. Whereas in the case where soluble E-Cadherin are present, so the soluble E-Cadherin will also compete for the sites that are present on the ECM for adhesions. So this will lead to the less adhesion sites available for the pancreatic cancer cells or PANC1 cells, and this will lead to the more invasions. And when they come up, when they check it for the aggregates, since this aggregates is also related to these adhesions, they are able to see that in the case where soluble E-Cadherins are present, they are able to see more adhesions. They are able to see more aggregations, not adhesion. They are able to see more aggregations compared to the case where there is soluble E-Cadherins are present. Because when soluble E-Cadherin are present, the similar logic that we have seen in the when we are discussing panel B, that when the soluble E-Cadherins are present, they will compete for the so these experiments are done on a single suspension cells. So the similar logic will not apply because the cells are suspended. So when the soluble E-Cadherins are not present, so since the chances of aggregates formations are higher, because the soluble E-Cadherins will then disrupt the 2 cells from interacting to each other, as the soluble E-Cadherins can binds to the can affect the binding of the cell aggregations. That is seen in the case where soluble E-Cadherins are present. So overall, they want to conclude that the soluble E-Cadherins which can be found or which are found to be high in the pancreatic cancer cells, because in pancreatic cancer cells there could be a high expression of this HK7 cells, which leads to the cleavage of this extracellular part of the E-cadherin. So in the soluble E-cadherin can helps pancreatic cancer cells towards invasions, at the same time less aggregates formations. So this is what they finally conclude from this paper. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.